The overall goal of this procedure is to measure longitudinal changes in the mechanical properties of the extracellular matrix in three-dimensional cancer models. This is accomplished by first harvesting cancer cells and growing the tumor spheroids by an appropriate method until they have reached the desired size. The second step is to embed the tumor spheroid in the collagen or other appropriate extracellular matrix. Next, the grid of sample points is constructed and a video is taken at each point. The final step is to analyze the video data and compute and co-register rheological properties at each spatial point. Ultimately, this protocol uses analysis of tracer probe trajectories to longitudinally quantify spatial changes in extracellular matrix mechanics in a non-destructive manner.
The idea is that by bringing together imaging-based measurements of extracellular matrix rheology with in vitro 3D tumor models that restore extracellular matrix interactions, this approach can provide quantitative insight into time-dependent changes in the mechanical microenvironment that accompany tumor growth, invasion processes, and response to treatment.
The method shown in this video adopts an agarose overlay approach to generate non-adherent 3D spheroids from the established pancreatic cancer cell line PANC1. To begin, mix 10 milliliters of cell culture grade water with 0.1 gram of agarose to obtain a 1% agarose solution. Heat the agarose solution to above 70 degrees Celsius before aliquoting 40 microliters of the solution into each well of a 96 well plate. Incubate the plate.